using the IDEAS beamline and looked at how the common earthworm impacts selenium speciation in contaminated soil. They were also our winners of the 2019 Judges Poster Competition and the Public Vote Competition. So they're really excited to share what they learned.
Yeah, so hi everyone, I'm Collegiate and we're the Evan Hardy Synchrotron group.
I'm Catherine.
I'm Elijah.
I'm Sebastian.
I'm Uday.
I'm Hannah.
I'm Eugene.
Ryan.
I'm William.
And I'm Chana.
Good morning all. I'll be introducing the topic of our Synchrotron research project, an analysis of selenium in earthworms. Selenium is an essential element for both humans and animals for multiple reasons, such as the regulation of metabolism, DNA production, thyroid gland function, and prevention of diseases. However, selenium exhibits a narrow range where it changes from vital to toxic for the body, so monitoring your selenium uptake is crucial for health. In soils, selenium is commonly found in its inorganic states of selenite and selenate. It also exists in its organic states of selenomethionine and selenocysteine, which are more bioavailable. This increased bioavailability means that the selenium is more easily uptaken and used by the body. Lastly, selenium can be used to remediate arsenic, as the two share an inverse relationship in regards to their concentration. As selenium concentration goes up, arsenic concentration goes down.
Our primary inspiration for this project was a study carried out on the CLS by a team led by Dr. Chaudhry, which also included Dr. Corbis. In this study, they exposed pregnant female zebrafish to environments with high concentrations of selenomethionine to mimic the runoff from coal and other metal mining. In this study, they found that selenomethionine readily replaced other methionines in the embryo, especially concentrated near the eye. They found this using imaging. This use of imaging is what inspired us to also use imaging in our project. And from this, we developed our question: can terrestrial organisms, such as Acina foetida, also uptake and change the speciation of selenium?
For our procedure, we began by purchasing Acina foetida worms. We divided 20 worms into 5 groups of 4. Each group was placed into a container with 48 grams of soil, 2 grams of shredded newspaper, and some distilled water added. To 4 of our containers, we added types of inorganic selenium as well, 2 concentrations each of sodium selenate and selenite. Our last container had nothing further added and served as a control group. The worms were kept for 2 weeks while being fed watermelon and shredded paper to keep them happy and healthy.
After these 2 weeks, the worms were euthanized using 4% buffered formalin. As seen in the top picture, we prepared our worms for the synchrotron. 1 worm from each container then had XRF or X-ray fluorescence scans carried out on it to look at concentrations within the worms. We also did XRF on the beddings from the 2 highest concentrations of selenium, as well as the control. We then did X-ray absorption near a structure known as Dynes on worms from the 40 milligram per kilogram selenite and selenate, which were higher concentrations, as well as a worm from the control group. So this looked at speciation within the worms, and these techniques will be elaborated upon in our results and analysis.
So, moving into our analysis, our first analysis used X-ray fluorescence, or XRF. So essentially, this hits samples with a beam, and that indicates what elements and the concentrations of those elements that are in our worms. So the XRF of Acina foetida shows that they readily uptake selenium in greater quantities when exposed to higher levels of it. Now, if I may direct you to this graph we have up here, it depicts the absorbance of selenium in the selenite-treated worm. So we can see that the control one, represented by the black line, had very little selenium in it. However, the 20 milligram per kilogram worm, represented by the red line, had elevated levels of selenium, while the 40 milligram per kilogram worm, represented by the blue line, had even more selenium than its counterpart. So the second graph shows the absorbance of selenium in the selenate-treated worm. It yields similar results as the selenite worm, but to a lesser degree. The selenite-treated worm reached an absorbance of over 10,000, while the selenate worm had only 6,000. So the worms exposed to more selenium took up a significantly higher amount of it. We can suggest that Acina foetida is capable of uptaking selenium in increasing concentrations upon exposure.
We also use the Dynes to determine the changing speciation within the worm. Dynes, which stands for X-ray absorption near edge structure, it uses normalized photon counts to plot the lines. As we can see in this graph, lines colored with purple and black represent the worms treated with sodium selenite and sodium selenate, respectively. While the middle two lines represent the inorganic standards, and the bottom ones represent the organic standards. As we can see in this graph, if we draw a line, the peaks for both worms move away from the inorganic peaks, which are colored with orange and blue, and it lines up with organic standards, which are colored with green and red.
So another thing we observed while we were looking at our XRF data was as the instances of selenium went up in our samples, the arsenic was reduced. Therefore, we used the IDS beamline to image the instance of arsenic in our samples. So Evan Hardy is actually one of the first couple of student groups to use the IDS beamline for imaging. And as you can see, the top images are control condition, whereas the middle and the bottom images are 40 milligram per kilogram selenite and selenate conditions respectively. And so the warmer values indicate a higher instances of arsenic, whereas the cooler values indicate a lower instance of arsenic. And as you can see, in our control condition, the arsenic is mostly situated towards the center, whereas this travels towards the back of the worm in our experimental conditions. Moreover, as the instances of selenium went up, the arsenic was reduced. This suggests that the both elements are inverse in nature.
Based on the analysis my peers have just discussed, there are 3 suggestions we were able to make about our research. Firstly, we were able to suggest that our worms were actually able to uptake the selenium we introduced into their system, and this was evidenced by our XRF data. Secondly, based on our Dynes data, we can suggest that the worms were able to change the inorganic selenium to a more organic form. More specifically, these forms were selenomethionine and selenocysteine. Thirdly, we observed an inverse relationship between arsenic and selenium. When you increase the concentration of selenium, the concentration of arsenic in the worm decreases. And this was evidenced by our imaging results. Based on these 3 suggestions, we can propose a way our worms can be used in Saskatchewan.
Alright, so moving on to the application of our work, you might already know that Saskatchewan accounts for approximately 20% of all Canadian cattle, and approximately 87% of prairie cattle have less than adequate levels of selenium. Combating this deficiency is obviously extremely important, but a major problem is that the organic selenium supplements, which are used to treat this deficiency, are extremely expensive. With our work, since our worms can naturally convert inorganic selenium to organic selenium, we hope to use them as an inexpensive alternative. Additionally, worms are already used as an excellent source of proteins for animals.
Looking towards the future, if given the opportunity, we'd like to further examine the sodium selenite and selenate treated soil without the presence of the worms. We would do this to look at whether or not the worms are responsible for the chemical change found in our results. Along with this, we would like to track the route traveled by the selenium in the worm to look at specifically what changes the speciation of the selenium. In our experiment, we use selenium. However, it would be nice to look at if the worms can take up other commonly deficient elements such as zinc, copper, iodine, and then change the speciation as previously done in our experiment.
This is the poster we competed with in the 4 different votes, and you can see it details all the different sections we talked about today.
We would like to acknowledge that the CLS is situated on Treaty 6 territory and the homeland of the Métis. We thank Dr. Chaudhry, Dr. Nichols, Dr. Corbis, Dr. J. Dynes, Anna Maria Beechler, and David Muir from the CLS for their continued support on our project, and thank you to our teaching supervisors, Mr. Yu and Mr. Kimberly, and all other Evan Hardy and CLS staff for their support. Thanks for listening. Any questions at this point?